Okay, I needed a while to cool off because it actually pissed me off the fact that it wasn't recording any of my audio from the past 45 minutes that I've been doing this. So I'm restarting every single thing at 1 a.m., which is my fault. I should have checked the audio. I should have done this yesterday. There's a lot of stuff I could have done. It doesn't matter anymore. Okay. So I don't even know where to begin anymore. We have myofilaments, we have sarcomeres. And then within the sarcomeres, so this whole thing right here, this is a sarcomere. And we have a repeating structure of sarcomeres that make up the muscle. And it's pretty predictable. We have an alternating areas of dark and light, and then these dark areas are going to be referred to as our A bands, which is going to be right here, and that's mostly in the middle. And then you also have like your H zone right here. So within the A band is the H zone, and then the I bands go basically from the end of each Z disc to the beginning to the end of each Z disc, which is the I band, which is referred to as our light band. The I stands for basically isotropic, the A stands for anisotropic, and then both of these terms refer to the light absorbing character of each of these bands. But yeah, so we have our Z line, or we could say these are the Z discs, and then the Z lines are divisions between the adjacent sarcomeres, and then the sarcomeres are connected end to end along the entire length of the myofibril. And then in the middle, again, of these A bands is our H zone, which means heli, or I guess bright. And then each A zone has a darker M line, which I don't really know how to show right here, but this would be like our M line, which is running down the middle of the A band. So we could do M line. And then if we kind of zoom in more into this area, this is what it's going to look like. So we could say that right here, this is the I band. Again, I band if there were more. And then this is our A band right here in between the two. Wait, is that right? No, that's not right. I don't know what I'm thinking. Sorry, let me fix this real quick. So, I band, A band. But you get the point. This would be the A band, this would be the I band. I band. And then in between here is where you're going to have your M line. And then these 2 points right here are going to be your sarcomere. So this is one single sarcomere. This is another sarcomere within the muscle. Muscle fibers are made out of myofibrils. So right here, so these like little like caps right here, this is going to be our cap C, and then these are our Z discs, basically, our Z discs. And then when we connect all of these together, we are going to basically form a alpha actin. And then right here, all of these like little filaments right here, this is going to be referred to as our F actin. And then at the little ends right here, this is our tropomodulin, which I kind of showed right there. So tropomodulin. And then we also have actin, and actin is going to be shown, not actin, sorry. I meant my, what am I talking about? Okay. F actin. And then here is where we're gonna find, so this right here is the titin. And then we have these like little heads, which is the myosin heads. And then at the end of basically right here is what I was talking about. This is our tropomodulin. And then around here is when we're gonna have these like F-actins, which are composed of G-actin. So these right here are G-actin molecules. And then I literally did it so perfect the first time. That's why I'm so pissed off right now. Each actin is composed of 2 strands of F-actin. So this whole thing right here is G-actin, F-actin, F-actin, 7 little monomers. And then you have a series of troponin and tropomyosin. So each F-actin is formed by 2 strings of G-actin. And then the G-actin has a binding site, which is like these little parts right here. Has a binding site for ATP. And then these binding sites which is basically a binding site for myosin. And then tropomyosin extends over the entire F-actin filament and covers myosin binding sites. Tropomyosin covers all 7 G-actin. And then tropomyosin, so calcium binds to troponin, moves tropomyosin off of the active binding sites, which reveals the myosin heads, and then they can bind. But troponin, I don't care anymore actually. Yeah, so troponin moves tropomyosin. So troponin has 3 factors to it. It has I, which binds to the active binding sites. ATP binding sites. That is not right, Sophia. Okay, I binds to G-actin. It binds to actin. T binds to tropomyosin. C binds to calcium. 2 low affinity, 2 high affinity. Lower intracellular calcium means that it basically has a high affinity, which helps to maintain the stability of troponin complex. And then an increase in intracellular calcium concentration means that we have a low affinity, which means a conformation change of the whole entire complex. And with this comes troponin pulling off the tropomyosin away from the myosin binding active sites. So then we can imagine this being basically off of it, revealing the thick filaments composed of 300 myosins, which are type 2 molecules. And then and they're also bound to titin, which we're going to kind of like show right here. So we have titin, we have nebulin. I don't know if I showed you that here, but this is nebulin. And then we have the titin, which wraps around tropomodulin, myosin, all that. Okay. And then when we're talking about myosin, this is kind of what it looks like, but it's also this little thing right here. So titin, the tropomodulin, myosin heads, but that is basically what this is. 2 heavy chains, 2 light chains. It's a myosin type 2. We have the neck region right here. These are the tails, and then this is the hinge region. This is the binding sites. So 2 light chains, 2 heavy chains, and then ATPase, which is going to be involved in the cross bridge formation. Because ATPase, it's basically hydrolyzing ATP, going from ATP to ADP and an inorganic phosphate. Yeah, so we have our little myosin head right here. And that's that. So that is us going over the molecular characteristics of contractile filaments found within the functional unit of the skeletal muscle cell, the sarcomere. And now we're going to go ahead and describe the action potential in the sarcolemma that results in the release of calcium in the sarcoplasmic reticulum, followed by a description of molecular mechanisms of muscle contraction. I guess it didn't take me that long. I don't know how long this is actually taking me now. Oh, no, come back, please. How long is this? Oh, it's not going to tell me. That's fine. I don't care anymore. Neuromuscular junction. Are you so excited? I'm so excited. I'm actually pissed off now. Okay. So, let me take a little sip of my drink. Amazing. 10 out of 10. It's old. Ew, don't look at my notifications. Okay. So, the whole point of this is to cause a muscle contraction. Calcium equals muscle contraction. Love. Anyways, so we have an action potential that arrives at the axon terminal of the somatic motor neuron, which connects the muscle fiber via the neuromuscular junction, or basically the synapse. So we have an arrival of the action potential, which causes voltage-gated calcium channels in the axon membrane to open, and then calcium's going to go ahead and open another one, and it's going to enter into the axon terminal from extracellular. And then calcium is going to bind to the snare proteins, and then that's going to cause an exocytosis of acetylcholine from the synaptic vesicles into the synaptic cleft. And then acetylcholine is going to go ahead and bind to these ligand-gated cationic channels, which are also known as your nicotinic type 1 receptors in the sarcolemma of the postsynaptic tissue, which is basically the end motor plate of the end motor plate. And then the binding of acetylcholine causes these sodium-potassium ligand-gated channels to open, which moves sodium down into the cell, and potassium diffuses out due to driving forces. And then sodium is going to basically depolarize the sarcolemma, opening more of these sodium channels, which initiates a action potential to spread out from the neuromuscular junction down the T tubules, which are filled with extracellular fluid, which are high in sodium, low in potassium. And then acetylcholine, we're gonna jump back to this right here acetylcholine, if it wasn't terminated, the signal would continue to induce action potentials. So we don't want that, because that would mean continuous muscle contractions or spasms. We have acetylcholine esterase, which breaks down acetylcholine in the synaptic cleft. I can't remember which one goes which, but I'm pretty sure it's acetyl that goes back into it's choline. Choline is taken up by the axon terminal and recycled to produce more acetylcholine. And yeah, it's just recycled so you can produce more acetylcholine, so we can continue this process of binding and causing action potentials down the sarcolemma. And then we're going into the T tubules. These are the T tubules. They surround both sides of the sarcoplasmic reticulum. Yeah, so we have action potentials going down these T tubules, which causes a change in potential. And then other voltage gated channels in the T tubules are going to respond to this change in voltage by opening up a channel called DHP, I can't remember the actual name for it, dihydro something, and then ryanodine, expressed as YRY. DHP is mechanically linked to ryanodine, which is ryanodine is going to be located on the sarcoplasmic reticulum membrane. And then this whole change in membrane potential due to sodium depolarizing a cell and causing more channels to open is going to make DHP open ryanodine, which is allowing calcium to leave the sarcoplasmic reticulum into the sarcoplasm. From the sarcoplasm into the sarcoplasmic reticulum. And then calcium binds to low affinity binding sites on troponin C, which causes it to move tropomyosin off of its active binding sites onto each actin G molecule, which is what we are basically referring to right here. So, uncovering, so let's imagine that this is being uncovered now, and it is, basically not vulnerable, but now it can do things now that it doesn't have something weighing it down. So uncovering active uncovering active sites means that our little myosin head is going to bind to actin. And then the binding of this is what causes our cross bridge formation. And then in its relaxed state, so this is when it is cross bridge, and then when it's in its relaxed state, it looks like this. This is what it looks like when it's in its relaxed state, because the myosin head is now, oh my gosh, what am I saying? Okay, so this right here, this is our cocked position because it's upright. Which means that it has high energy. Cocked is equal to high energy. And then it's recaptured in the phosphate bond, and then actually we'll move up here real quick. So this is when it's in its high energy state, but not able to bind to actin active sites because it has ADP and inorganic phosphate attached to it. So that's basically what's happening right here. This is when it is in its cocked position because we have the tropomyosin basically being removed off of it. So calcium comes in, it moves, calcium binds to troponin, moves tropomyosin off of the active binding sites, and then this is when it's in its relaxed state. The head is cocked, it's high energy, but it's not able to bind to actin active sites because it has ADP and an inorganic phosphate attached to it. So troponin binds to calcium, and that moves tropomyosin off actin active sites, and then myosin binds to actin, and then that initiates a bond that releases energy, and then this is when the bond is broken and it releases energy in the form of heat, or I guess it escapes as heat. And then we have the power stroke, which is the removal of this phosphate. So yeah, right here, removal of this phosphate, this inorganic phosphate, which causes the myosin head to basically hinge or become uncocked. Returning it to its low energy state, which is basically pulling actin past myosin. And then ADP is also released in this. So we have ADP and an inorganic phosphate released, which kind of leaves it with nothing, because that's what causes it to be in its low energy state. The fact that it has nothing attached to it, it's just vulnerable, ready for its next step. So releasing the myosin from the actin causes it to go into this recovery stage. So ATPase. So we have ATP, which is basically coming in, and then we have ATP, and then the creation of this bond is going to release heat. So we have ATPase here, which basically hydrolyzes. So ATPase hydrolyzes ATP into the inorganic phosphate and the ADP, which causes it to become recocked, and it prepares it for the next power stroke. So it goes cross bridge formation, power stroke, release, recover. Cross bridge formation, power stroke, release, recover. I don't know why I put that there. Anyways, where did my other little thing go? Okay, so now we're gonna come back to this little state, and I want to give notice to the fact that this only, wow, did I just erase that like an idiot? This only applies to skeletal muscle, because now we're going to go ahead and talk about smooth muscle, which is basically when we could use an example of peristalsis, when you're moving your food from your mouth and your esophageal tract and then into your intestines to be digested. The main difference, oh my gosh, how long has this been going on for? Because I'm over it. I don't know how long this has been going on. Okay, girly pop, we're almost done with the first part of this essay. Smooth muscle can shorten 80% of its length. I don't know what it is for skeletal muscle, if I'm being honest, but the main difference between this one and skeletal muscle is that fact that we are using these little proteins, I think, or monomers, which are called MLCK, and then we have rho kinase, which are pretty cool. So the same process occurs. Titin, Nebulin, tropomyosin, all of that. These are the myosin heads, hinge, tail, head. We got that? Amazing. So we start off with the phosphate and ADP. This is a sarcomere. It's thin, has no troponin on it. It has no troponin on it because calcium came in, bind into troponin, move tropomyosin off. I'm pretty sure that should say tropomyosin. But yeah, so calcium comes in, and then we have MLCK. I don't remember exactly what that stands for, but then we also have ADP added in as well. ADP and the inorganic phosphate, this is when we are going to have a high affinity for calcium, and then we're going to basically break this bond, generate heat. The phosphate's going to come off, ADP's coming off, this is our power stroke, and then we're going to turn into here where there's an inorganic phosphate, and then we have ATP coming in. ATP is generated through this bond, and then that creates heat. But MLK, in the presence of calcium, is going to turn on MLCP. MLK. I know I was I don't know what's wrong with me anymore. MLCP, MLCK, I'm pretty sure, yeah. So calcium, because we have a high affinity right here for calcium, MLCP is basically an enzyme that is going to eat at our MLCK, and which turns off Rho kinase. And turning that off means that we basically stop this because we don't have enough calcium in order to be able to start this phase over again. So sometimes because of this, it'll just slowly continue, but it'll take its time and hydrolyze and contract and go straight over to the power stroke initiation of the cross bridge cycle, which is kind of nice because then you don't have to worry about ATP having to break into ADP and an inorganic phosphate. It already just occurs. Instead of, it still hydrolyzes, that's the thing, but you don't have to worry about this power stroke because you can just go from cross bridge, power stroke, release, or you can go from recover straight to power stroke. And then that is for phasic. For tonic, the only difference is, so we still have all of that going on, MLCK, MLCP, all of that. But right here we have this latch phase, which is when it stays contracted without burning any type of energy, like the energy that we created here, creating this bond of ATP. And then here, yeah, so it's ADP and then an inorganic phosphate instead of just ATP, because ATP can't bind to something that's already got ADP and an inorganic phosphate. So this is when we're just taking our time to continue to contract. Okay, and now we're going to talk about so we're going to talk about various sources of energy utilized for muscle contraction. And then we're going to talk about the relative amount of each source available to the cell, and then the advantage and disadvantages of each type, under what conditions each might be used from. And I'm going to quickly go ahead and glance at this because these are the different types of muscle fibers that we have, but we're going to talk about energy sources first. So energy sources that could be used would be our creatine phosphate. So creatine, we think of bodybuilders, but we actually produce creatinine in our bodies, which is then converted into creatine phosphate. So creatine phosphate, we could think of this as being like an immediate source of energy, and then it basically donates a phosphate group, so ADP plus inorganic phosphate, which creates an ATP. And then the advantages of this ATP means that it's fast. It's a fast, reliable source. The only disadvantage of this is it would be the fact that it's limited. It's a limited supply, and then it's used during short bursts of energy. I'm so sorry. My handwriting is going to be so bad right now. So we could say this is our first one, and then second one would be glycolysis. Good old glycolysis, specifically anaerobic process. Because glucose is not glucose. So we have glucose, right? It's broken down into pyruvate and then into lactate because of the absence of oxygen. And then some of the advantages of this would be is that it rapidly produces ATP. And then one of the disadvantages would be that it's inefficient because you need 2 ATP per every glucose, which then causes lactic acid to build up, I think. Or is it lactate? No, it's lactic acid. I don't know anymore, actually. Lactic acid to build up. And then, yeah, so we could just say lactic acid. And then, let's see. This would be used during, like, a moderate length or high intensity exercise. So, high intensity, because, you know, at, like, some point you do, like, have to have the stamina, the metabolism, not the metabolism, what am I saying? Stamina. I think stamina is the only word I want to use right now. And then 3, we could say aerobic respiration. And then that's when we have glucose, fatty acids, amino acids, and these are all going to be metabolized with oxygen in order to create ATP. And then some of the advantages of this one would be that we have a high ATP yield, so, you know, 36 to 38, which makes it sustainable. And then a disadvantage of this would be is that it's a slower onset, because we have to create more ATP for every ATP that we do, like, get rid of. It's a bit slower. And then for this one, you could say that we use it. That's awful, actually, sorry. For low intensity workouts. So, used in low intensity workouts, which we could say endurance, like running. And then this one, high intensity, for that one we could say sprinting. I mean, not sprinting, sorry. This one would be like 30-second interval efforts. This one would be for sprinting. That's what I was meaning to say. Yeah. So, I don't know what I'm talking about anymore. Those are going to be our various sources of energy utilized for muscle contraction, and then each source available, and then the disadvantages and advantages of each one. And then we already talked about the 2 main types of smooth muscle, which were our phasic and our tonic. We already talked about that. That was my bad. I didn't mean to jump ahead in that. And then describe the molecular mechanisms. Okay. So the only thing that is left for us to do is to compare and contrast smooth muscle and skeletal muscle structurally and functionally, and the regulation. I already did that. Okay, and then describe and explain, and then compare and contrast the muscle sensory receptors, spindles, and the Golgi tendon organs. We have different muscle types, and I'm just going to go ahead and slide over here because I am not going to rewrite it. I don't have the energy to do that anymore. It's almost 2 a.m. I have church at 9 a.m. So that means I have to wake up in like 5 hours, 6 hours, whatever. My gosh. Okay. So, actually, let's go ahead and go over here. So this is when we are going to be talking about comparing skeletal, my gosh, skeletal versus smooth. We already talked about tonic, phasic within smooth muscle, and we also talked about skeletal muscle and the contraction of that. But so we could say with skeletal muscle, they're both skele, smooth. Let's go ahead and draw that line down. We could say that with skeletal muscle, this is voluntary. And with smooth muscle, it's involuntary. Nuclei. We have multi-nuclei. It's multi-nucleated. This one is single, which would make sense because we need a lot more oxygen in order to be able to produce the actions that we want to with skeletal muscle. And then striations, we have striations here, no striations here. And then we have sarcomeres, which are present here because it's muscle. So sarcomeres, no sarcomeres for you. And then calcium. So calcium here, calcium comes from the sarcoplasmic reticulum, and then calcium here comes from extra cellular sources. And then the proteins that are used for skeletal muscle would be troponin. And then this one would be calmodulin. And then, so we have like fast ones here, and then we have slow, and they are rapid, fatigue resistant. But we're also going to go ahead and head back over to this little model that we have, because we have type 1, type 2, type 2X fibers, which are within the skeletal muscle. So we have type 1 is going to be slow, and we get our calcium source from the Surca pump within the sarcoplasmic reticulum, which is slow acting. And then we have troponin as our regulatory protein. And with type 1 muscle fibers, they're small in diameter. We have myoglobin, which is high because we want more oxygenated red blood cells in order to create the energy needed. And it has a high capillary disc. We have the type 2A, which is fast, and it has the Surca pump pumps out calcium fast for this one. Its regulatory protein would also be troponin. It's medium diameter. Its myoglobin is high, and then its capillary disc is medium. And then we have the type 2X, which is fast as well, Surca pump, which pumps calcium out fast, troponin. It has a larger diameter, but it has a lower amount of myoglobin, and a low glycolytic process. Fatigue and not responding. So with type 1, we are fatigue resistant. Same with type 2. So with these ones, you could say that this would be, let's see. So fast twitch, so these slow twitches with high myoglobin, this would be when you want to be a sprinter because you're using up all this energy really fast, and then it's not, sorry, that's not the right one. So fatigable, this one would be when you're sprinting. This one right here, type 2A, would be fatigue resistant, because this is for people who run marathons. Their power output would be medium, because with marathons you want a high amount of myoglobin so that you're able to maintain this output of, what's the word, maintenance, I don't even know, of like being able to run. But with this one, type 1, you would see it more in females, because we it's just more of like a longer stride, able to go longer. And then you have a type 2X, which is seen more commonly in males. Alright, it's the same day, it's just not 2 a.m. It's actually 8:30 p.m. and I'm doing this now. So we have a muscle spindle. Oh, the fact that I have to re-explain all of this again is pissing me off. We got a muscle spindle, right? Muscle spindles, they're located within the muscle fibers, and their function is to be able to detect change in muscle length, or stretch, we could say. And the structure, so we have intrafusal fibers, which are going to be inside right here. And then we have our extrafusal fibers, which sit on the outside. And then, let's see, when you activate either of these intrafusal fibers or these extrafusal fibers, they're going to lead to a reflex contraction of stretched muscle, which is basically your stretch reflex. And then within our muscle spindle, we also have voluntary control movements that depend on upper motor neurons. So upper motor neurons, UMN, upper motor neurons, and then we also have our lower motor neurons. And then the cell bodies of the upper motor neurons, those are in the cerebral cortex, where you have things like your planning, initiation, and coordination movements. And then we have the upper motor neurons, the upper motor neurons also are the neurons that synapse with the lower motor neurons of the spinal cord. The lower motor neurons, so these are going to leave the spinal cord, and then they synapse with the skeletal muscle at the neuromuscular junction. And then so we have our alpha motor neurons, and then we also have our gamma motor neurons. And these are important because so what it is that we have a receptor. Let me go and go over here now. So say we have a receptor, and that goes to the dorsal root ganglia of the spinal cord. So this is our little spinal cord right here. This is going to be known as our control center, and then we have this little outside part, which is basically the spinal cord. So we have our dorsal root ganglia, our ventral root ganglia, and then right here we could say that this is our receptor, which is going to receive the stimulus. So stimulus happens right here, our receptor notices it, and that causes an action potential down this neuron. And since it's going to the control center, this one right here is going to be our sensory afferent neuron, meaning that's going to the spinal cord. So afferent. And then we have one that goes out from to like produce our executive function, and this one is going to be what produces the response. So this one is going to be the efferent because it's leaving the spinal cord, and then this is basically going to be the effector. This is our motor neuron right here. So yeah, there's that. I feel like that's all I really need to explain for the part where it says to describe and explain the function, and then compare and contrast the sensory receptors, muscle spindles, and then the Golgi tendon. So receptors, we have numbers of receptors. So one receptor would be, so a receptor, let's start with that. What is a receptor? It's something that detects a change in stimulus, and then it'll react. So we have a mechanoreceptor. Mechanoreceptor, think stretch. It senses stretch or a different type of like mechanical pressure or disorientation. And then we have chemoreceptors. We have talked about this. Chemoreceptors, they sense a change in certain chemical stimuli. And then we have our thermoreceptors, which these ones detect temperature and a change in temperature. And then we have our photoreceptors, and these ones are in the eyes. So these are going to be able to detect it's specifically in your retina, I think, and these respond to light. And then we have our nocioreceptors, and nocioreceptors, these are the ones that detect a change in like pain. These are your pain receptors. Okay, so yeah, we went over afferent, control center, efferent, then the effector. So you also, within the control center portion of this reflex, so this whole thing right here, this is what makes up our reflex arc. And then simple reflexes involve like a single synapse between two motor neurons. That would be monosynaptic. So again, monosynaptic is simple reflexes involve a single synapse. So single synapse between two neurons. And then polysynaptic, this one would be when we have two or more synapses. Two or more synapses. So we have different types of reflexes. We have a somatic reflex. We have a stretch reflex as well. Okay, and then I want to go over this one more time just in case I wasn't like too clear on it. So this right here, this is going to be our intrafusal fiber, and then what wraps around these intrafusal fibers would be our muscle spindle. So our muscle spindle, this is what wraps around it's an afferent sensory technically, meaning that we are going to the spinal cord. And then on like basically the ends of these like little spinal muscle spindles, we have our gamma efferent motor neurons. And what these gamma efferent motor neurons are doing is, oh no, I forgot. Hold on, let me look at my notes real quick. I don't know where it is in my notes actually. Oh, okay. So the gamma motor neurons, they are the resultant of contraction of a spindle. In response, gamma stimulation is too weak and it contributes to gross muscle movement. And the reason that this is important because of the sensitivity of the muscle spindle while the muscle is being shortened or lengthened. And the sensitivity of these spindles is because of the stretch contraction. The gamma motor neurons can be divided into, hold on, let me skip over here. So we have our gamma motor neurons, which can be divided into bag as well as chain. So these two muscle spindles, they respond to stretch, but the bag fibers we are going to say are associated with type 1A sensory neurons. And then they're most sensitive to responding to a stretch. So most sensitive. And then we have our chain fibers, and then they're slow. So we can say that these ones are slow and sensitive to length of time. Sensitive to length of time the stretch is occurring. Yeah, so that's what we have for intrafusal, and then we're going to get into our stretch reflex. Let's go ahead and microphone on. Okay, good, thank goodness. All right, so the stretch reflex. This one is going to be when, let's see. It's like when you transfer one weight to the other, so that like in the case of, I think the book used like you're falling or something like that, or you're sliding and you use one foot to catch yourself so you don't fall from under you. So what you have is you have the muscle spindles, and those ones are going to send a message to your alpha motor neurons to the spinal cord. So we have this right here, the what am I saying? So it's going to happen right here. It's your abductors, and then the muscle spindle is going to sense a change in stretch. So one, I guess, change in stretch, and then that's going to go ahead and synapse with the alpha motor neuron, and then the alpha motor neuron is going to cause the adductors to contract. So alpha motor neurons, alpha motor neurons, when they're stimulated, they're going to inhibit the hamstrings. So inhibit hamstrings. And then so this is the leg. This is what I'm trying to get at. This is the leg. This is the hamstring. This is the quads. So your sensory receptor neuron conducts an action potential, which is basically going to be this, and this is going to go up here and synapse with this guy. But this is when it's going to go to the spinal cord, or the brain, the control sensor, and then the sensory neurons are going to synapse with the alpha motor neurons and the interneurons. So alpha motor neurons and neurons, and then that was my bad. Yeah, so we start here, and then we go from the sensory neuron conducts an action potential, which then goes to the brain, to the spinal cord, the control center, and the sensory neuron is going to synapse with the alpha motor neurons and the interneurons up here in the dorsal root ganglia, and then we travel down to the ventral root ganglia, where the alpha motor neuron to the quads is stimulated, and the alpha motor neuron to the hamstrings is inhibited because we're trying to contract. So we'll just use these arrows. So quads, and then this is when the quads are going to be stimulated and contract, so that way you can balance yourself. They contract, these relax. That is the end goal of this stretch reflex. And then we also have our, let's go over here, our Golgi tendon organ reflex, and this is where muscle spindles respond to stretch in another type of sensory system. but responds to tension. So we can think of stretch and tension being the same thing, but they're really not. I'm so sorry. I kind of forgot that this music was going. Let me go ahead and pause it real quick. I just don't know how to like focus without it. So hopefully that didn't distract you too much. Okay. So, yeah. So our Golgi tendon reflex, it's going to be kind of what helps us balance. And it's what's, I think what the book uses is that it like keeps the bone from being able to tear off or from its attachment site. It's keeping everything together in an orderly manner. The stretch reflex can be excitatory and cause the contraction of the stretch muscle group, and the Golgi tendon reflex could be considered inhibitory and cause relaxation of the affected muscle. So then we don't have as much tension, and it's to help us to be able to prevent injury. And it helps us in regulating muscle contraction force. So the Golgi tendon organ the Golgi tendon organ signals muscle force through the entire physiological range, not only at high strain. So it's going to be the same exact process. We have step 1 here. The Golgi tendon organ senses excessive tension. Right here, this is where we are feeling the excessive tension. Sorry, not there. This is where we would be feeling the excessive tension right here, near the patella, and then it's going all the way up here to the dorsal root ganglia, and this is when the sensory neuron conducts an action potential, and we go to the dorsal root ganglia, and then this is where we're synapsing, and we're going to the control center to be able to tell the body what to do in order to prevent the tension and tendons from detaching. So the sensory neuron synapses with whatever, I don't care anymore. Okay, so sensory neuron synapses with interneurons, not alpha motor neurons, just the interneurons. And then right here is where it's going to connect here and here, and then we're going over to the hamstring. That's the ultimate goal, so that we can prevent the tension. But right here, the alpha motor neuron, so the quads are going to be inhibited. So quads are inhibited, and then the hamstrings again are stimulated. So the overall goal is to work on the hamstrings. So hamstrings contract, and then the quads relax. And it's not the same in every situation, but that's what's the main difference between the Golgi tendon organ and the stretch reflex. And then we have the withdrawal reflex over here. So the withdrawal reflex is kind of like if you're standing on a nail and you need to take your foot off, so you bounce your weight to the other side. So the cross extensor reflex, instead of like hurting that foot, you're putting the weight onto another one. So this one has excitatory and inhibitory interneurons on the same side of the body, and the pain neuron also sends a branch to an excitatory interneuron that crosses over the opposite side body of the spinal cord. So what this one is going to look like is, so say this foot, this is the nail, right? Ow. Okay, so we want to balance it to this other foot, the weight, I guess. So what's going to happen is the overall effect of this is we're going to relax and then contract here. And this is for the foot that is being affected. So we have our stimulus right here, the stimulus, right? And then it's going to go over to the dorsal root ganglia. So stimulus, dorsal root ganglia. We're going to synapse with the not intermotor neurons. What am I saying? Ow, jeez. Oh, I did this last night and I don't want to do it again. Okay, so the flexor muscles are going to be stimulated to contract, and then the hamstrings and the hip flexors, once you step on the tag, it's going to excite. And then you're going to just move your weight to the opposite side. So your flexors contract and the flexors also relax. So your flexors on the foot that got stimulated, the flexors on that foot that got stimulated are going to contract so that you can move this leg in order to, so once this one's done contracting, this one over here is going to contract, but for now it's relaxing. So we're still going to have the same process. We go here, we synapse with the intermotor neurons. Intermotor neurons, and then we have these little guys right here, and then we're going over to the other leg down here, and we'll be contracting here and relaxing here.